In this video, we're going to be discussing the electron transport chain. So before we can get into the details of the electron transport chain, we need to understand the structure of the mitochondria. So this is my sketch of the mitochondria. Right here we have the outer mitochondrial membrane. Here we have the inner mitochondrial membrane. In between them, something called the intermembrane space, which is going to be very important to our understanding of the electron transport chain. And deep inside, we have the mitochondrial matrix. Now, located on the inner mitochondrial membrane, there are going to be these proteins or complexes that are going to be responsible for the process of the electron transport chain. So it's important to know that the electron transport chain is going to take place on the inner mitochondrial membrane. Okay, so let's just understand the electron transport chain in very simplistic or in a very simple way before we enter the details. So that way we have an outline of what needs to be understood. So we said before that we get NADH and FADH from the TCA cycle. Now, these are electron carriers, and they're gonna give their electrons to these complexes that are located on the inner mitochondrial membrane. Now, each time one of these complexes gains an electron, it's going to enter a state or a high energy state because it gained an electron. And this high energy state allows them to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. So after a while, we're going to get an accumulation of all these hydrogen ions inside the intermembranous space. So what's going to happen is that they're going to gain an electron, and they're just going to keep passing these electrons on to the second complex, to the third complex, and to the fourth complex. And in the process, they're going to be pumping all these hydrogen ions out into the intermembrane space. So at the end of this process, what you're going to have is you're going to have high concentration of hydrogen ions in the intermembrane space, and low concentration of hydrogen ions in the mitochondrial matrix. So we have a gradient that's going to push hydrogen ions from the intermembrane space into the matrix. So how does all this generate ATP? Well, we have a protein that's called the ATP synthase protein, and it's going to synthesize ATP from the movement of hydrogen ions from the high concentration in the intermembranous space into low concentrations in the membranous matrix. So the more hydrogen ions that move from high concentration here to low concentration here, the more phosphorylation of ADP that you're going to get, and the more ATP that's going to be produced. So in a nutshell, the electron transport chain is the process of synthesizing ATP via a process that is called oxidative phosphorylation. So why is it called oxidative phosphorylation? Well, because there's an oxidation part that happens in the complexes. So every time they lose an electron, this is an oxidative process. Oxidation is the process of losing an electron. And there's also a phosphorylation part in which there is phosphorylation of ADP into ATP. So the last thing that we need to discuss before we start discussing the details of the electron transport chain, we need to understand what happens or what metabolic pathways take place before the electron transport chain begins to function. Well, like we said in another video, we discussed the process of glycolysis, in which glucose is converted into 2 molecules of pyruvate. In this process, ATP and NADPH are going to be generated. So you get 2 ATPs and 1 NADPH molecule. Now, pyruvate is then going to be converted into acetyl-CoA inside the mitochondria by the enzyme pyruvate dehydrogenase. After we get acetyl-CoA is then going to enter the TCA cycle and produce multiple NADH and FADH2s. Now, these are going to be the electron carriers that go and enter the electron transport chain in order to produce ATP. Okay, so now we're going to begin to discuss the specific processes that happen inside the electron transport chain. And so we're going to start by building this model right here. Now, I know it looks a little bit complex, but just bear with me through this video. So right here we have the outer mitochondrial membrane, and right now we have the inner mitochondrial membrane. So we're going to start with complex 1 because this is where the electron transport chain starts. Now, complex 1 is also called NADH dehydrogenase. Why is it called that? Because basically it's going to oxidize NADH. So it's going to take the electron from NADH. Now remember, we said before that NADH and FADH are electron carriers. So complex 1 is then going to take this electron from NADH and give you NAD+. Okay, so the electron is going to go from NADH and it's going to go to a specific structure on complex 1 that's called flavin mononucleotide, also abbreviated FMN. The flavin mononucleotide is then going to give its electron away to something called iron-sulfur compounds. Now, when complex 1 has that extra electron or has gained an electron, it's going to be in a high energy state. And when it's in a higher energy state, it's going to have the power to pump ATP molecules from the mitochondrial matrix into the intermembrane space. So complex 1 is characterized by this little protein right here that can just take these hydrogen molecules or hydrogen ions and pump them into the intermembrane space. So we're gonna begin to see hydrogen ions accumulate in the intermembrane space. Okay, so after the pumping is done, complex 1 is going to send its electron all the way to something called coenzyme Q. Coenzyme, okay, so this is the end of complex 1. Now we're going to talk about the role of complex 2. So complex 2 is also called succinate dehydrogenase. And what it's going to do is that basically it's going to oxidize FADH2. So FADH2 is going to be converted to FADH, and the electrons that it lost is going to be taken in by FMN that is located on complex 2. Now, complex 2 does not have a protein that could pump hydrogen ions into the intermembrane space. So what it's going to do is that it's just going to give its electron to coenzyme Q. So just to sum things up, NADH is going to be worked on by complex 1. FADH is going to be worked on by complex 2. They're both going to take those electrons and eventually give them to coenzyme Q. So now we've reached a state in which the coenzyme Q is going to have all the electrons. So what it's going to do is that it's going to give these electrons to complex 3. Complex 3 is also called cytochrome BC1. Now, when complex 3 gains this electron, it's going to enter a high energy state, and that's going to allow it to pump hydrogen ions from the mitochondrial matrix into the intermitochondrial space. So after it's done pumping, complex 3 is then going to give its electron to cytochrome C. Cytochrome C is then going to give that electron to complex 4. Complex 4 is also called cytochrome A plus A3 or cytochrome C oxidase. Now, of course, when complex 4 gains that electron, it's going to enter a high energy state and it's going to start pumping all these hydrogen ions. Now, the last step in the electron transport chain is that complex 4 is going to give its electron to oxygen. Now, when an electron binds with oxygen, in addition to hydrogen, it's going to form water. And this is going to be the last step of the electron transport chain. So at the end of the electron transport chain, we have an intramembrane space with high concentration of hydrogen ions, and we have a matrix with low concentration of hydrogen ions. So how does this help us in the production of ATP? Okay, so the difference in the concentration that is formed leads to an electrochemical gradient from the intermembrane space into the mitochondrial matrix. Now, as hydrogen ions are moving from the intermembrane space into the mitochondrial matrix through the protein ATP synthase, this movement of hydrogen ions allows for the phosphorylation of ADP and the synthesis of ATP.